Good morning, everybody. We are going to be continuing with chapter 8. Great. Yes, I'm doing fine. Quick question. Did we do the beginning of chapter 8 not last week, the week before the break? I seem to remember doing it, but I'm not 100% certain. Did we do genetics? Yes, wait. Do you remember me doing anything on genetics? Have you started filling in your notes on Mendelian genetics? Just wanted to. Okay. So I'm trying to remember where we left off. You take a look really quick. If I can remember where we left off. I think we left off on incomplete dominance. So either way, I'm going to go ahead and start there. Okay, so hopefully we'll have some more folks joining, but let's go ahead and get started. Okay, from 2 weeks ago, when we did this, we talked about the basic concepts of dominant and recessiveness, how you have one gene from each parent. The dominant always shows, and the recessive only shows if there's two of them. Okay, so what we're going to be doing now is talking about a different kind of characteristic, and if I've already gone over this, then use this as a way to just get back up to speed. Okay, so this type of characteristic is called incomplete dominance, and the reason why it's called incomplete dominance is because one color doesn't cover the other; they blend together, which is kind of logical. So, for example, red is not dominant over white, which you would guess. Instead, they blend together. They're equally dominant, or what we say co-dominant. So, or incomplete dominance. Now, there is a little bit of a difference between co-dominance and incomplete dominance, but in this case, let's just focus on incomplete dominance. And that means when you have two very different traits, they blend together with each other, and you see the blended phenotype. Okay, so in incomplete dominance, the F1 hybrids, that means a blend between the two extremes, have an appearance that's between the two phenotypes of the parents. So red and white makes pink. It's logical. So neither one is dominant. Don't assume red is dominant just because it's the darker color. How's this different from normal dominance or recessiveness? Now, if red were dominant to white, if you had a gene for red and a gene for white, the flower would be red instead of pink. Now, we use a different set of symbols for this. And because it's incomplete dominance, everything is capitalized, but instead of a uppercase and lowercase letter, we use an uppercase letter with an uppercase superscript. So in this case, it means color red. So this flower has two genes for red color. Color white. This flower has two genes for the white. Now, you know it's not hiding anything because whatever is in there shows up. In incomplete dominance, nothing ever hides. It always shows. So if you only have red to give, then this red flower is going to only give the gene for red, color red. So it's a different notation. This flower can only give color white, so you only show one box. So what happens is all the offspring are pink, which kind of makes sense. But now if you look at this level, the pink flower can either give a red gene or a white gene. So half of the gametes they produce are red, half the gametes are white. This is still a heterozygous condition, one of each. Now, if you put these on the Punnett square, you see red only comes first because it's first in the alphabet. Try to remember that. In incomplete dominance, we list them in alphabetical order, not dominance to recessiveness. Red is not dominant to white. And the same over here. So when we fill them in, we see that red comes back when two red genes fill in. We see pink when white comes down and red comes down. Another pink over here from the other combination, red and white, and then white. So when you mix the two pink, you get all the colors out. This is how incomplete dominance works. Okay, multiple alleles. Multiple alleles is different because of this. Now, in all the traits that we've been talking about before, there was dominant and recessive, red and white. There was only two versions of every gene. Now, in this case, multiple alleles, there's going to be three or more. So, for example, there's a gene for O, a gene for A, and a gene for B. That's three different alleles, different versions of the gene for blood type. Okay, so if you look at this figure here, you can see blood type O is shown by double recessive I. Now, why is it I? It stands for immunoglobulin. Immunoglobulins are these proteins that stick out of the surface of a red blood cell that could cause it to clump together. You guys have probably heard you have to receive the correct blood type or you'll die. Why do you die? Because if you put the wrong blood in someone, it clumps up, their blood can't pump. And if your blood is solid, you will die. Okay, so blood type O, which is interestingly double recessive, but more people have it. No one knows for sure why. So two little i's meaning double recessive. Now A is dominant. So if you're blood type A, you can either have big IA, which shows dominance, big IA, so you can have two of them, which makes you type A, or you can have big I and little I, big IA and little I. So in this case, the O gene is recessive and so it hides. The same thing for B. B is dominant, so you can either have double B or big B, little I. Either of these will be. But the interesting thing is these are what is called co-dominant because A and B, if they're present, will both show up on the surface of the red blood cell. So if you have a gene for A and a gene for B, you're type AB. If you're not A minus or B plus, both things show. So it's AB. Okay, let's do the notes. The ABO blood groups are an example of multiple alleles because there's 3 or more alleles. 2 of the human blood type alleles exhibit co-dominance. Which ones are they? A and B. They're co-dominant. Now, in incomplete dominance, remember they blend together to form an intermediate color. In this case, co-dominance means they both completely and fully show up. For example, in cows, there's a co-dominance in color where you have red patches and white patches. So you see both red and white. The cow doesn't come out pink. It comes out patching red and white. So you see both. All right, both the alleles are expressed in the phenotype, and you'll need to be able to do these problems in lab. So we're going to be doing this in lab. I'll be asking you to solve genetics problems using these allele markers. All right, another topic here is called polygenic inheritance. So what does polygenic mean? Well, the prefix poly means many, and genic means genes. So it means many genes working together to give a single look. In this case, there's 6 spots on the chromosomes that when you add all the color genes together, and as a matter of fact, each spot is kind of an incomplete dominance because every time you add a gene for darkness, you add a little bit more color. So for example, if you find somebody here up at the top of this that has 5 genes for light skin and 1 gene for dark skin, you would have a very light brown individual. But down here, if you have all the genes for dark, you would have a dark black skinned individual and then all the combinations above. So if you found somebody who had intermediate brown skin and they married somebody else of intermediate brown skin, you could have children all the way from completely pale to completely dark, depending on what combination of these genes were passed on, because at each spot, you can either give the light or the dark, light or the dark, light or the dark. So these are all the combinations of gametes that a person can give for skin color. Polygenic inheritance is the additive effects of 2 or more genes on a single phenotype. So the idea is instead of just having 1 gene from mom and 1 gene from dad and then use that, there's actually 3 pairs here, 3 pairs of genes. Every time you add a little bit more dominance, you would think of it, the more color you get in the skin. Now, this is also variable based on tanning, but for base skin color, this is basically how it works. The more copies of the dominant traits, it's kind of unfortunate that they use the term dominant. It should be the more copies of the colorful gene the person receives, the more dominant or the more darkly colored they appear. Skin color in humans is a good example of that. And this graph means that when you mix these 2 people together, you're going to get mostly similarly colored children, but you might have a very pale skinned child and maybe a very, very dark skinned child from these 2 medium brown parents. All right, sex-linked. We're going to talk a lot more about sex-linked in lab, so I'm going to go through this relatively quickly. Sex-linked has to do with the fact that these traits are carried on the X chromosome, and they're not carried on the Y. And as a matter of fact, what I'm going to do is I'm going to go through this in detail in lab, so I'm not going to spend a whole lot of time talking about it here in lecture, because unless we can do some problems, unless I can show you how it works, it doesn't make a whole lot of sense So let's continue with the notes. Okay, a number of human conditions result from the sex-linked or X-linked genes. That means that they're found on the X chromosome, which is a sex chromosome. Red-green color blindness. Now, if you were red-green colorblind, you would not be able to see the number here. If you see the number 7, it means you are not red-green colorblind. It's characterized by a malfunction of light-sensitive cells. This is kind of a little bit discriminatory. It's just a different way that people look at the world, not the same as most people. Okay, so let's see. Have I stopped sharing? Let's see. What is going on here? Why? It's not giving me any options. Huh. Oh, there we go. Okay. All right. Meeting controls is on a separate thing. Okay, so let's see. Do we need to do chapter 8, part 2? Oh, no. Wrong thing. All right, so that is it for genetics. So we're going to believe it or not, most of what I'm going to be teaching you about in genetics will be in lab. So you do need to come to lab next week. That's when we're going to be doing genetics problems. And then after that, we'll be doing human variation. So we have a lot of time talking about genetics. Most students really find this fun because it's all about us instead of talking about cells and animals. And most people are mostly concerned with themselves. So we're going to be talking about how do we inherit certain characteristics from our parents and pass them on to our kids. All right, Bella, let's see, what do we have? Do we have anything happening? Nope. So there's no test and no lab, right? Yeah, there's no online lab, so we're going to be, what did we do Monday? What did we do Monday? Oh, we did the walk around campus. That's right, plant stuff. Okay, so we're going to be doing genetics problems starting on Monday, and then the following week we do human variation. So it's a little bit off from what it says on the Canvas page, but just come to class and you'll be good to go. Bella, do you have any questions before we call it a day? Okay, well, thank you for coming, Bella. I hope this was helpful, and I hope to see you on Monday in lab, and have a great week and a great weekend. See you later.